Hi everyone, and welcome to our summary video all about the ultrastructure of eukaryotic cells. This particular video we're going to be focusing on the membrane-bound organelles. We'll have a look at the other organelles in a future video. So in terms of the specification, this is looking at the section 2.1.1g, which as you can see there is the ultrastructure of eukaryotic cells and the functions of the different cellular components and the list of them that we are looking at during this video. Before we delve into the actual organelles themselves, it's always worth reminding ourselves of what eukaryotic cells actually are. So what we have is any cells of animals, plants, fungi, or the protoctistans are eukaryotic cells. So quite simply, if they ask you about any of the features of a protoctistan, then you can just jump straight on in by saying they are eukaryotic cells. And the key feature that we learnt back in GCSE about these eukaryotic cells and the difference between eukaryotes and prokaryotes is that the eukaryotic cells have a nucleus surrounded by a nuclear envelope that contains the DNA. Now, what we can see here is just how much at GCSE and at Key Stage 3 we just kind of lied to you about what cells look like. I mean, gone is the animal cell that literally just had a little shaded nucleus in the corner and that was apparently it, maybe a few dots if you were lucky. We can see on the left what the animal cell actually looks like. Here it is. So this is just a generic animal cell if you like. And what we can see labelled on there are all these key features that we're going to be looking at throughout this video and future videos on our ultrastructure. So some of the ones that we're going to look at today, we've got our nucleus, and we're going to look at the nucleolus as well, which is that little darker region within the nucleus itself. We're going to be talking about these nuclear pores and the nuclear envelope. We're also going to talk about the endoplasmic reticulum, both smooth and rough. We'll talk about the Golgi apparatus, or Golgi body as it's labelled in this diagram. And we're also going to be having a look at the mitochondria. Now, if we look at the one on the right over here, we've got our plant cell. So again, we lied lower down the school. It wasn't as simple as we led you to believe. So we can see that we do have some of those common features, cell wall, cell membrane, etc. But we're also going to be looking at these ones that we didn't mention previously. The Golgi apparatus, your chloroplasts. We're going to be looking at the mitochondria, the vacuole, etc. So one thing that you need to be able to do in terms of your A-level papers is to recognise these different parts on these diagrams. So what I'd suggest you do is print off a copy of the pictures. You can find these on numerous sites on the internet, and then just kind of print it out without the labels, or just cut the labels off. And then just practice labelling what those things are. And actually, the more of these images you look at, so the greater the number of different diagrams you look at, then the better you're going to get at being able to identify them in an unfamiliar diagram on an exam paper. Before we look at the specifics of individual organelles, let's actually look at what an organelle is. Now, organelles are these subcellular structures, these things that occur within cells, and each organelle has a very specific function. The reason that we've got these organelles is that by having all these different little units within the cell that have their own specific function that they carry out, that means that we can actually divide up all those jobs that the cell has to carry out to each of those individual organelles. And that means it's going to be way more efficient than just basically having a sack with everything to do everything contained and kind of hoping that it all works itself out in the end. By using these organelles, each one with a specific function, we can divide the labour up, we can make it way more efficient at carrying out those tasks. What we're looking at in this video are the membrane-bound organelles. Now, as the name suggests, it's not a lie here, these are the organelles that have a membrane around it. And this creates something we refer to as compartmentalisation. Now, if we talk about compartmentalisation of a cell, what it does is it's separating out those individual bits needed for a particular function into a given region. Hence compartmentalisation. We're putting things into little compartments. And what we actually find is that these membrane-bound organelles are only found in eukaryotic cells. So we previously referred to just the nucleus, but more generally, membrane-bound organelles are only found in eukaryotic cells. Let's get stuck in then with the specific membrane-bound organelles that we're going to look at. And number one on our list is the nucleus. I'm hoping that from our earlier studies we do know that the nucleus has kind of three key functions really. First one is it controls the cell, so it controls all the activities within the cell there. Secondly, that's where the organism's genome, and remember the genome is the full genetic code of the organism, is stored. And it's going to provide those instructions that are necessary for that process of protein synthesis. So those are our kind of three key features of what the nucleus actually does. What we're going to do then is we're going to have a little look at the individual structure. So we're going to break it down into little sections here, and then the function of that particular structure. So we're just on the nucleus at this point in time, and the first part of the nucleus we're going to look at is the nuclear envelope. Now, we don't say that the nucleus has just a membrane. We describe it as a nuclear envelope because it's a double membrane, okay? Now, what that means is that we've got those two layers running around the outside of this nucleus to form that envelope. And actually going through the nuclear envelope itself, we've got these little things called nuclear pores, so little tiny holes running through, and we'll look at those a little bit later in this little video. Why do we have this nuclear envelope then? Well, it separates all of that nuclear contents from the rest of the cell, so it's keeping all of that genetic material in one location away from everything else in the cell. We will also find that on that nuclear envelope that the inner and outer membranes of it have fused together in some regions to actually allow ribosomes and dissolved substances to cross. Now, it only works in that way with smaller substances. If we need larger substances, this is where the pores come in. So what we see here is when we need to move things like the mRNA from the nucleus where the DNA is found to the ribosomes, which are going to be within the cytoplasm area, then obviously we need a method to do that because mRNA is too large just to fit through these regions where the membranes are fused. So nuclear pores allow these larger substances to actually leave the nucleus, or it allows certain things, and these steroid hormones you will look at in a later part of the course, to enter the nucleus. And that means those steroid hormones can then have a direct impact on that genetic material. The next bit of the structure we're going to look at is this region called the nucleolus. Now, the nucleolus is just a region of the nucleus, and it contains RNA. Now, don't be fooled into thinking that there's some kind of a membrane surrounding it, holding it all together in that region. There is no membrane around it. It's just a region. You'll be able to pick it out in images because it's this little darker patch on the nucleus. It's not always found in the middle, and this diagram here does have it sort of in the middle, but it's not always in the middle of the nucleus. So in terms of microscope images, you're looking for a little darker region, and even on the diagrams you can see it's just shaded in that little bit darker there. In terms of what happens in our nucleolus, well, that's where the ribosomes are made, and ribosomes are going to be very important in terms of our protein synthesis, which we'll look at later. The third key part of the structure of our nucleus is this material called chromatin. So we can see chromatin here, okay, these are the little wavy lines in the diagram. When we talk about chromatin, this is DNA that's been wound round these type of proteins called histones. Now, what we find is that depending on which stage of the cell cycle our cell is at, that will determine whether the chromatin is all condensed and therefore it's sort of coiled up into these chromosomes, or if it's not in the process of division, then it's actually all kind of spread out, and the best way to describe it is it's kind of like a tangled ball of string really. So what we find is the chromatin will only be condensed into those very clear chromosome structures when the cell is in that process of dividing. Other than that, it's not condensed. Key function of our chromatin, that's where the genes are. So that's where we get the genes for all the different characteristics of our animal. The next structure we're going to look at then is the rough endoplasmic reticulum, or RER. Now, when we consider what the rough endoplasmic reticulum is, if we look at the diagram on the right first of all, what we can see is it's actually this entire little system made up of these fluid-filled cavities. And we call these fluid-filled cavities cisternae. Those fluid-filled cavities, the cisternae, are actually continuous with the nuclear membrane. And if we have a look on the right here, you can see down in this bottom corner, so we've got our nuclear envelope, and then you see there's that little line, and then it goes straight into the rough endoplasmic reticulum. So it's continuous with that nuclear membrane. Why is it called the rough endoplasmic reticulum? Well, all these little dots around the outside, those are actually ribosomes. So because it's got the ribosomes associated with it, then it is the rough endoplasmic reticulum. In terms of function, it's got a large surface area, and that means there's plenty of sites for ribosomes to be attached. The ribosomes are going to be incredibly important because this is where protein synthesis is going to take place. So our rough endoplasmic reticulum is able to synthesize those proteins. Once they've been produced, they're then transported to a different region of the cell called the Golgi Apparatus, and the way that they're transported is through those cisternae. And you can see it's like a little grid network where you can just travel through them. The next membrane-bound organelle we are going to look at is the smooth endoplasmic reticulum, or SER. So again, this is our system of membranes containing the cisternae, which are the fluid-filled cavities that are continuous with the nuclear envelope. So it's the same in a lot of ways as the rough endoplasmic reticulum. The key difference, there are no ribosomes on the surface of the smooth endoplasmic reticulum. If you look down here, no little dots on the surface, no ribosomes present. So that's our key difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough has the ribosomes, the smooth does not. The function, however, is very different. So whereas the rough endoplasmic reticulum is the site of protein synthesis, the smooth endoplasmic reticulum contains the enzymes required for lipid metabolism. So this is the site of the synthesis of these various lipid molecules, cholesterol, the lipids, the phospholipids, steroid hormones, all synthesized within the smooth endoplasmic reticulum. It's also going to be involved in absorption, synthesis and transport of lipids as well, obviously transporting through those cisternae just like we saw with the rough endoplasmic reticulum and the proteins. Next up in our membrane-bound organelles is the Golgi apparatus, sometimes referred to as the Golgi body in certain texts and books. Now, what we actually have here is a stack of membrane-bound flattened sacs. And if we look at the picture, you can see that it actually looks quite similar to the endoplasmic reticulum. Now, this is where we need to be careful, because they can give you various diagrams or microscope images of the ultrastructure of cells and ask you to identify something that is basically a stack of membrane-bound sacs. The key thing to look for, is it attached to the nuclear envelope? If the answer is no, you have got a Golgi apparatus. If the answer is yes, it's a kind of endoplasmic reticulum. Then look for ribosomes or not. So make sure you don't mix up Golgi apparatus and endoplasmic reticulum. The other bit on this little diagram we've got are these little bits down here, little circles. These are secretory vesicles. Now they're basically little transport mechanisms that allows us to move materials between the Golgi apparatus and elsewhere in the cell. So what actually happens with our Golgi apparatus? Well, we've got two sides to it. The cis face is where these transport vesicles bring substances in, and the substance they're bringing in will be proteins from the rough endoplasmic reticulum. So those proteins are brought into the cis phase, and then once they're in the Golgi apparatus, they will be modified. So the key function of the Golgi apparatus is protein modification. And this can take different forms depending on what we're trying to make as the end result. If we add a sugar molecule to our protein, we make a glycoprotein, and this is important in our plasma membrane structures. If we add a lipid, we make a lipoprotein. It can also fold those proteins up into their three-dimensional shape, which hopefully from biological molecules section we know is the tertiary structure of the protein. So the Golgi apparatus is all about protein modification. Once it's modified the protein, on the trans phase, the one on the right there, this is going to package the proteins up into these vesicles, and then they will either be stored or transported to the plasma membrane. When they get to the plasma membrane, they will either fuse with it and then become part of the membrane itself, or be released to travel elsewhere. So what we have there then is our Golgi apparatus. The next membrane-bound organelle is the mitochondria. Now, mitochondria have a very distinctive rod shape, as we can see from the diagram, and they're about 2 to 5 micrometers long. They do have a double membrane that surrounds them, and the inner membrane there, if you look at the diagram, is very folded. Now, these folds of the inner membrane are what we refer to as cristae. We will look at the mitochondria again when we come on to respiration. So this isn't just the only time we see it, but it is a good chance to learn it now because it's going to then be built upon when we do the respiration. If you then look around those cristae, we've got this in the diagram blue stuff, and that is this fluid-filled matrix. So they can ask you to label bits on a diagram of the mitochondria. So cristae, matrix, those are two of the main ones to know at this stage. What does it do? This is the site of aerobic respiration. So this is where we're going to be generating ATP. Next structure is the chloroplast. So chloroplasts, hopefully we know, are found not in animal cells, but in plant cells and certain other organisms, but never in an animal, remember. And what we find is these are 4 to 10 micrometers long, so they're quite large. They have a double membrane. The inner membrane itself is made into these little stacks of flattened membrane sacs, and we refer to these as thylakoids. Within those thylakoids is the chlorophyll. So here's our little diagram in the bottom corner, and these little stacks that look like a stack of coins, those are the thylakoids. The stack itself is referred to as a granum. So each individual little disc there is a thylakoid, and the whole stack of them together is the granum. Around that we've got our fluid-filled matrix, that's called the stroma, and we will find that there's loops of DNA and starch grains within our chloroplast as well. What does it do? Just like we learnt lower down the school, it's the site of photosynthesis. We will obviously look at photosynthesis in more detail, but the key parts are traps the light energy using the chlorophyll, and that's going to be used to make ATP and split the water molecules. Hydrogen reduces our carbon dioxide in order to make carbohydrates within the stroma. Our next membrane-bound organelle is the vacuole. Now, we only find a large permanent vacuole in plant cells. Animal cells will not have this large permanent vacuole, so it's something that is only in plant cells. In terms of what it is, well, it's the membrane called the tonoplast surrounding a fluid. So basically we have this fluid inside this tonoplast, the membrane. In terms of what it's used for, this is what's going to fill up with water. So water moves into the vacuole in our plant cells. As it does so, it's going to swell out when those cells are turgid, and it then pushes against that cell wall, creating this turgor pressure, which creates that rigidity within our plant cells, allowing them to grow vertically without a skeleton. Our penultimate structure we're going to look at are the lysosomes. Now, the lysosomes themselves are formed by the Golgi apparatus, and they have a single membrane. Inside that single membrane, they have these things called hydrolytic enzymes. So basically, when you look at it, it is a circle, and in there we've got these hydrolytic enzymes. Okay, that's my amazing diagram for you. So single membrane around the outside, hydrolytic enzymes inside. Where do we tend to find lots of them? Phagocytic cells, so the ones that are involved in our immune system. In terms of why they exist, well, hydrolytic enzymes are actually quite powerful digestive enzymes. Now, we use these enzymes in order to break down sort of old organelles within the cell, any foreign material they encounter, the whole purpose being that we can then repurpose the bits that made those up for the cell to use in future constructions. It wouldn't be very useful if those hydrolytic enzymes were just free floating within the whole cell. In that scenario, any time they happen to come into contact with an organelle, they would digest it. So it wouldn't go too well for our cells to just be randomly having organelles digested willy-nilly. So instead, it separates those hydrolytic enzymes from the rest of the cell contents, so we will only digest the things we want to. We then come to our Cilia and Undulipodia. Now, quite simply, both these have similar structures. They are projections from the actual cell itself, which are surrounded by the cell surface membrane. They contain these structures called microtubules, and they're formed from another structure called a centriole. In terms of what they do, different things in different regions of the body. We've already heard about ciliated cells, I hope. These are the ones that make up certain parts of your airway, and it's all to do with moving the mucus. Another place you've hopefully heard of these is in our sperm cells, which we're going to upgrade to spermatozoon. Now, these have a structure we refer to as the undulipodium, officially. Sometimes you see it as flagellum. To be perfectly accurate, we should be saying undulipodium. And the whole purpose of it being there is to allow movement, because hopefully we know that in order to fertilise the egg cells, then those sperm cells have to actually get there in the first place. We'll also find in more recent research that every cell, or virtually every cell, has a single cilia projecting from the surface, and that is actually involved in cell signalling, so it's able to sense the environment around it and then know what's going on and therefore make changes as appropriate. Hopefully you found this video useful. Don't forget to subscribe to the channel so you get all the latest updates when we upload further videos in the future.